Hello everyone. We are continuing with the chapter of pollination and fertilization, and today we will do that will be E part of this pollination and fertilization. And in this part we are going to study about fertilization. As you have studied in the previous classes that fertilization is the fusion of male and female gametes. So here we will study this fertilization in a detailed way. Successful pollination is followed by fertilization. You have in the previous session you have seen how the transfer of pollen grains from anther to stigma takes place, and once the pollen grain is landed on the stigma successfully, fertilization or the process of fertilization starts. Fertilization is the fusion of the nuclei of male and female gametes leading to the formation of zygote or the oospore. Fertilization is the fusion of male and female gametes leading to the formation of zygote or the oospore. That means the result of fertilization is the formation of zygote, or it is also known as the oospore. So before that, starting studying the this process of fertilization, we should know the structure of pollen grain and the structure of ovule. So here we will study about the structure of pollen grain. The pollen grain is a microscopic and unicellular structure. See, pollen grains are very minute and they are very, very small in size, and they are unicellular structures. It is covered by two layers. I told you that a pollen grain is covered by a two layer. It is a two-layered structure. The outer layer is very hard and thick called the exine. So the pollen grain is surrounded by two layers. The outer one is very hard and it is thick and it is rough also, and it is known as exine. The inner layer called intine is thin walled. See, outer one is hard, it is thick, it is rough and is known as exine, and the inner layer is thin and is known as the intine. The exine at certain places has pores called the germ pores. So the outer layer, which is thick and hard, has many pores in it. Small pores are present in it, and these pores are known as the germ pores, through which the pollen tube emerges out during its germination. Now see, the exine has a small pores known as the germ pores, and through one of these germ pore, the pollen tube emerges out during the process of its germination. The pollen grain has two nuclei called the tube nucleus and the generative nucleus. And this pollen grain has two nuclei. One is known as the tube nucleus and the other one is known as the generative nucleus. So you can see here the structure of a mature pollen grain. You can see this is the outer layer. It is hard, rough, thick and is known as the exine. The inner one, you can see here, this inner layer is thin and is known as the intine. This pollen grain has two nuclei, that is, one is known as the tube nucleus. You can see here, this is the tube nucleus, and this one is the generative nucleus. So this is the structure of a pollen grain, a mature pollen grain. See again, this pollen grain is a microscopic unicellular structure. Now the outer wall is it is surrounded by a double layer. The outer one is hard and rough, thick, known as exine. This outer layer has many pores in it, and these pores are known as the germ pores, through which one of this pollen tube emerges out. Then the inner one is thin walled, it is known as the intine, and this pollen has two nuclei, that is, one is known as the tube nucleus and other one is known as the generative nucleus. So this is the, see here how the pollen tube is emerging out after dissolving this exine, or after the rupture of the exine, the intine develops into a pollen tube. See here, this one, it is growing. And see this nucleus, this is the tube nucleus. And these are the male gametes inside this tube nucleus, tube, pollen tube. So this is all about the structure of pollen grain. Now we will see the structure of an ovule. Ovules are present inside the ovary, and in the ovary, these ovules are attached to the ovary with the help of placenta. Now, structure of the ovules. The ovule is a small structure present in the ovary. It is a small structure which is present inside the ovary. It is attached to the placenta by a stalk. See, ovules are attached to the placenta by a stalk called the funicle. See, ovules are attached to the ovary by placenta, and it is attached to the ovules are attached to the placenta by a stalk called the funicle. See again, ovules are attached to the ovary by placenta, and ovules are attached to the placenta by a small stalk, and that stalk is known as the funicle. So, the funicle provides nourishment to the ovule. So, underline it, this children, this is the function of funicle. So, what is the function of the funicle? It provides nourishment to the ovule. Now, again you see, the ovule is covered by two protective layers called the integument. So, ovule is also surrounded by two protective layers, and these layers are known as integuments. In the same way as this pollen grain is surrounded by a double layered, or it by two layers, exine and intine, in the same way ovule is also surrounded or covered by a protective layers known as the integuments. It bears a small opening called micropyle at one end. See, these two layers, they bear a small opening at one end, and that opening is called as the micropyle. The ovule contains a structure called the embryo sac. So, inside the ovule is a structure called embryo sac. See, this is the diagram of ovule showing the embryo sac. See here, this is the diagram. This is a mature ovule, we can see. This is the funicle. This one is the funicle. That means this funicle, this ovule is attached to the placenta. See, this is the placenta. It is attached to the placenta by a stalk known as, this ovule is attached to the placenta by a stalk known as funicle. This funicle provides nourishment to the ovule. Now, it is surrounded by two layers, protective layers. They are known as the integuments. See, this is one layer, this one, and this is the another layer with which this ovule is surrounded, or it is covered. Inside the ovule, this ovule contains this embryo sac. You see, this is a sac. Here, you can see this is an embryo sac which is present here inside the ovule. So, this is the embryo sac. In the embryo sac, the following structures are present. You can see in the embryo sac, a female gamete called egg towards the micropyle. See, this one is the micropyle here. The opening in the two, between the two integuments, you can see here, this is known as the micropyle. And a female gamete, that is egg, you can see here, this one. 1, 2, 3. The center one is known as the egg. It is known as the female gamete. This one is the female gamete, or it is also known as the egg. Two polar nuclei which fuse to form the secondary nucleus in the center. Here you can see the diagram of an embryo sac. This is the egg, and these are the two polar nuclei. When they fuse, they forms the secondary nucleus in the center. So one is the egg lying towards the micropyle. In the center, two polar nuclei are present, which after fusion will form the secondary nucleus. This polar nucleus polar nuclei, they lie in the center. Two synergids. Now see here, with the egg you can see two synergids. Three antipodal cells. These are the three antipodal cells which are opposite, lying opposite to the egg, or we can say lying opposite to the micropyle. Here is the micropyle. You can see here, this is the micropyle. Here is the egg. Two synergids are there. In the center, two polar nuclei are there, and opposite to this micropyle, these are the three antipodal cells. So the embryo sac contains one egg, two synergids, two polar nuclei, and three antipodal cells. The embryo sac is surrounded by a nutritive tissue called the nucellus. Now see here, this embryo, this is the embryo and it is surrounded, you can see here, this one, it is surrounded by a nutritive tissue. You can see this nutritive tissue, and this nutritive tissue is known as the nucellus. And the function of the nucellus is to provide nourishment to this embryo sac. So this funicle provides nourishment to the ovule, and this nucellus provide nourishment to this embryo sac. So once again you can see here the structure of the ovule with embryo sac. Ovule is attached to the placenta by a stalk known as the funicle. Ovule is surrounded by two layers known as the integuments. The function of the integument is to protect the ovule. You can see these integuments. Inside this is an embryo sac. See here, one opening is there in the integument, that is known as the micropyle. Inside this ovule is an embryo sac with an egg, two synergids, in the center two polar nuclei, and three antipodal cells opposite to the micropyle. And this embryo sac is surrounded by a nutritive tissue, and this nutritive tissue is known as the nucellus. Its function is to provide nourishment to the embryo sac. Children, this diagram is very, very important, and this embryo sac also. The diagram of pollen grain also is very important. Practice it and learn all its parts. Now this is all about the structure of pollen grain and the structure of ovule. Now we will see how the fertilization occurs. What happens? What are the events in fertilization? What are the events? What happens during fertilization? What events takes place during fertilization? During fertilization, a number of events takes place which are summarized as follows. See, during fertilization, many events takes place, and now we'll see what are these events. After the pollen grains are deposited on the stigma, they absorb nutrients, water and sugar secreted by the stigma and swells up. Growth of pollen tube is stimulated. Now you can see here, this is the ovary, this is the style, and this is the stigma. And you have seen the pollen grains have landed on the stigma. Now, once the pollen grains are deposited on the stigma, they absorb the nutrients, that is water and sugar, which is secreted by the stigma, and these pollen grains, they swells up. Once they swells up, what happens? Now the growth of the pollen tube is stimulated. You have seen this is a pollen tube. The growth of the pollen tube is stimulated. See, this pollen tube is growing here. Now, from one of the germ pore in the exine, the intine of pollen grain grows out into a fine tube called the pollen tube. Now see here, this what happens? See, this exine is ruptured, it breaks, and the intine growing into a pollen tube. So once the growth of the pollen tube is stimulated, what happens? This exine from the germ pore, in the exine, the intine of the pollen grain grows into a fine tube that is known as the pollen tube. The pollen tube grows and passes through the solid style. See here, this style is solid. It is very, very solid, but you have seen this pollen tube growing, passing through the solid style. How it happens? Now see, the tissue of the style are dissolved with the help of enzymes secreted at the tip of the pollen tube. Now see this pollen tip of the pollen tube, it is secreting some enzymes which dissolves the tissues of the style and thus helps in the growth of the pollen tube. Eventually, the pollen tube travels down the whole length of the style into the ovary. See here, the pollen tube grains, they are taking the nutrition from this stigma. They absorb the nutrients secreted by the stigma, and through one of the germ pore, the exine ruptures and the intine grows into a pollen tube. You can see here this tip of the pollen tube contains some enzymes which dissolves the tissues of the solid style, and thus it grows into, or we can see the tube travels down the whole length of the style. Meanwhile, the generative nucleus divides by mitosis into two male gametes or nuclei. We have seen this in the pollen grain that it has two nuclei. One is the tube nucleus, another is the generative nucleus. When the pollen tube is growing, what happens? This generative nucleus divides into two male gametes by the process of mitosis. Now, the male gametes move to the tip of the pollen tube and lie in the mass of the cytoplasm. See here, this is the pollen tube, and inside this is the mass of cytoplasm, and this male gametes. These are the male gametes. They lie. That means the generative nucleus is divided into two male gametes, and they are lying in the mass of the cytoplasm. And this is the tip of the see here at the tip, tube nucleus is there, which leads to the growth of the pollen tube, which helps in the growth of the pollen tube. Now, the tube nucleus degenerates. Once the generative nucleus has divided into two male gametes, now the male gametes are lying in the mass of the cytoplasm. After that, the tube nucleus, it degenerates. The pollen grain with a pollen tube containing two male gametes constitutes the mature male gametophyte. Now, what happens after that? This tube nuclei or the tube nucleus, it degenerates. This tube nucleus degenerates. Now, what is present? The pollen grain with a pollen tube and two male gametes. It constitutes a male gametophyte. So, a male gametophyte is without the tube nucleus. The pollen grain tube carrying the two male gametes turn towards the micropyle and enters the embryo sac. Now see here in this diagram, you see here the pollen tube is growing. It turned towards the micropyle. See, here is the micropyle, the opening between the integuments. Micropyle, it turns towards the micropyle and enters into the embryo sac. So, this pollen tube is carrying now the two male gametes. That means the generative nucleus has divided into two male gametes, and now this male gametophyte is ready to enter through the micropyle into the embryo sac. This is the embryo sac. The pollen tube carrying the two male gametes now turn towards the micropyle and enters the embryo sac. Now tip of pollen tube dissolves. Now once it enters, after reaching here the embryo sac, the tip of the pollen tube dissolves. it dissolves and it enters into the embryo sac. Now, the two male gametes are released into the embryo sac. The tip of the pollen tube is dissolved here, and once it is dissolved, the two male gametes are here. It enters into the embryo sac. One of the two male gametes fuses with the egg. You see here in the center here, you see this diagram. This is the egg. The pollen tube has entered here. The tip has dissolved, and this tube is carrying two male gametes. So one of the male gametes, it fuses with the egg. See here, this diagram. It has entered, and one of the male gamete with fuse with the egg cell and to form the fertilized zygote. So one male gamete fuses with the egg to form the zygote. The second male gamete fuses with the secondary nucleus or the definite nucleus. It forms the endosperm nucleus. The secondary nucleus is formed by the fusion of the polar nuclei. We have seen here two polar nuclei are here. When these two polar nuclei will fuse together, it will form a secondary nucleus in the center. And one of the male gamete will fuse with this secondary nucleus to form the endosperm nucleus. Have you understood, children, now? What is there? One male gamete is fused with the egg to form the zygote. These two polar nuclei, they fuses to form the secondary nucleus. Now the another male gamete will fuse with this secondary nucleus to form the endosperm nucleus. The zygote develops into an embryo. The zygote, which is formed by the fusion of the egg and one of the male gamete, will form the zygote, whereas the endosperm nucleus develops into endosperm. The endosperm nucleus, which is formed by the fusion of the another male gamete and the secondary nucleus, now forms the endosperm. And the function of the endosperm is to provide nourishment to the developing embryo. Embryo is formed by that zygote is formed that by the fusion of the egg and the one of the male gamete. That is zygote. And now this endosperm will provide nourishment to the developing embryo. So children, this diagram is also very important. Fusion, it is showing the fertilization. See, these are the synergids. These are the integuments here. This is the egg cell which will fuse with the male gamete to form the zygote. These are the polar nuclei which forms the secondary nucleus, and this secondary nucleus will again fuse with the one of the male gamete to form the endosperm nucleus. Now, what is double fertilization and triple fusion? In angiosperm, the two different types of nuclei in the embryo sac, egg cell and the secondary nucleus, are fertilized by the two male gametes released by the pollen tube in the embryo sac. This is called the double fertilization. See, double fertilization is that one of the male gamete is fusing with the egg. So that is one fertilization, and two of the polar nuclei or the second after fusion they forms the secondary nucleus, and that secondary nucleus fuses with the another male gamete to form the endosperm nucleus, and that is the second fertilization. So this fertilization is known as the double fertilization. The fusion of the second male gamete with the two polar nuclei is often referred to as the triple fusion. Now, second male gamete is fusing with the two polar nuclei. So two polar nuclei fuses with one male gamete, that is referred as the triple fusion. So these two definitions are very important. What is double fertilization and what is triple fusion? Now see here. Synergids do not seem to be essential for the process of fertilization. So there is no role of synergids in fertilization. They get degenerated after fertilization or even before it. So these two synergids which lie on either side of the egg cell, they degenerate after fertilization. So there is no this they are not essential in the process of fertilization. In case the egg cell fails to be fertilized due to any reason, the synergids also called help cells assume the role of egg cells. Sometimes the egg cell fails to be fertilized by the male gamete due to any reason. Maybe there is no fertilization or fusion between the male gamete and the egg cells. So these synergids, they are also known as the help cells, assume the role of the egg cells. Sometimes they also help thus they are known as the help cells, and they play the role of the egg cell. Antipodal cells also do not have any positive role to play and may disappear even before fertilization. See these three antipodal cells which are lying opposite here, they also do not play any role in the process of fertilization, and they this disappear even before or after fertilization. So here we end this session, and tomorrow we will see these are the events which are you have seen in taking place in the fertilization. Children, read it again and again. Prepare the diagram. Very important diagrams are there. Functions of each and every part you have to study. Double fertilization, triple fusion, very important, children. And tomorrow we will see the post fertilization changes in the ovule. So thank you and bye.